you all. All right, you see the full screen here? Yes. Okay, so hello everyone. I'm so happy to be here with you today, and thank you for the invitation and reaching out to me about giving a talk, as well as joining this chapter of the Native Plant Society, and for being here tonight. So again, I'm Maya, and I'm going to be telling you about this cool desert shrub called Glossopetalon, and I studied this for my master's research. And that was done at Northern Arizona University under the guidance of Dr. Tina Ayers. And this talk will be a little bit of storytelling, some science, and just really sharing with you all how beautiful this experience was for me. So to introduce you to these shrubs, the scientific name for Glossopetalon is just that, and it means tongue petal in Latin. But the common name is greasebush. But, you know, a lot of people haven't heard of this shrub, so you don't really hear it referred to very much by its common name. And the reason why they call it greasebush is because when those stems are wounded, sometimes there's this dark looking sap that comes out. And so that's how it's gotten that common name. And when I came into doing this work, there were 4 species in this genus. So a genus is just going to be that grouping of species. And 3 of those were endemic. So they were found in a very small restricted area and nowhere else on Earth, and were of conservation concern. And so those are listed for you here. So Glossopetalon cochlei, this is found in Nevada, and then in some mountain ranges in California. Texense is found in Texas, as the epithet hints, and that's along the Uvalde River. And then Glossopetalon pungens is also found in California. So these were really restricted taxa. And the last species in this group is Glossopetalon spinescens, and this is super widespread. And this is the species that you can find here in New Mexico. And so this species is so widespread that there's different populations along the range that look quite different depending on their environment. And so because of that, botanists have come along and named those populations just so we don't get confused. And that's what we call the varieties. It's just describing that variation throughout the species range. And so there's 6 of these names within this species. And if you're looking for Glossopetalon, I'm going to teach you how to identify it and tell you all about what I found out about this group. But when you're out looking for it, it loves limestone cliffs and outcrops. And so here are some pictures of the habitat. Here, this is in Nevada. That's my knee. And so when I was hunting for this shrub, I was up on this ridge. You can see the trees down below. And so you are in some some interesting places when you can find Glossopetalon. Here's another example along this gorge here. So there's some down here, they're dormant. And you can find it a lot of times along arroyos and gorges. But if you have limestone substrate, you have a good chance of finding this particular group of shrubs. And they can be growing straight out of the rock. So here, I'm sitting here and in the back of me, I have that cliff there, and then it's growing straight out of there, just straight out of the rock. And so that's what you really want to be looking for in terms of your environment in thinking about, well, can I find this shrub? And so here, this is a distribution map that I created of the genus's range. And so you can find it throughout the Western US and into Northern Mexico. There's a lot less points here just because we haven't had as much sampling done in Northern Mexico. So hint, hint, there's some opportunities there if you want to contribute to understanding this group better. And then you can find, we're going to talk specifically about the taxa found in New Mexico, and I'll explain how to identify them. But here's those points for the endemics that are found in Nevada and in California. And then you can also see that other species, Texense, that's only found here in Texas. And so these other colors with the var in front of them, that's all spinescens. So spinescens goes, this particular species goes really, really far throughout the Western US and into Northern Mexico. So you may recognize this type of map. This is from SciNet, which is that great portal that we can look at herbarium vouchers. We can see where things have been collected. And this is what we can find here in New Mexico. So aridum, I kind of colored them the same. Aridum is that, wow, it is really throughout the species range. So that's going to be that variety. You can find that here. Meonandrum, so that's kind of up in this corner. And then Planetarum, that's our purple dots, and then spinescens. And so we'll walk through each one of these, and I'll teach you how to identify the specific varieties within this species. So this species is going to be really variable. That's why we have varieties, but basically what distinguishes it from the other three in the group is going to be its height. So it'll be taller. It has stipules, and you'll see a really good picture of that on the next slide. And these are just these little like apertures that protect the apical meristem as the leaf develops. Oftentimes they can become thorny. So you'll see that like in mesquite, for example. What color is the petiole? So that bottom portion of the leaf, and then they have these swollen bases here that are just crazy. And then whether or not it's deciduous, so whether or not it just loses its leaves, that's kind of how you tell this species. And then I'll tell you, based on my experience and the key that I wrote, how I tell the varieties apart. So before I did this work, and honestly, even after, it's a little bit hard to tell these varieties apart. So for that widespread variety, for aridum, these are going to be your big leafy shrubs. And so they're very large compared to the other varieties. They have huge stipules. And so there's that stipule. You can see that adonate portion of the petiole. So like here's our petiole, and then this base, it's all swollen. And then this is our stipule. So aridum's going to have these stipules, and then they're going to have very big leaves compared to your other varieties of spinescens. And then they're also going to be widespread. So if you're thinking about just probability wise, you're probably looking at aridum if you're throughout the Western United States, but there's some considerations there. So that's that particular one. You're looking for something big, leafy. So as big as up to your waist, that's how large they can get. And then this one can also be found in sandstone. So here you can see down here. So this picture was taken in Sedona. This was the first member of Glossopetalon I learned, but I never even remembered learning it as a master's student, because what I saw was this when I learned it. So I had just moved from Alabama, never been to Sedona. I saw this and the plants. I was like, oh my goodness, look at all of those red rocks. Look at the moon up here. And I was blown away. And so my advisor taught this course. This was Native Plants of Arizona, this was the course that we were learning this group in. And I was just like, wow, look at that. And then so she was so kind. She's like, well, let me get a picture with you. You can share it with your mom. And she got a picture of our entire class. And so it was a bit overshadowed in that moment, how cool this shrub that I would go on to SETI was. But I'll tell you another story of when I saw it again, and it really sparked my interest. So in addition to aridum, we also have variety spinescens. And so whenever you give a scientific name to something, you have to have it based on a particular plant or specimen. And so you'll see some redundancy in naming where the type specimen, so that one that the name is based on, you can use that same epithet as the species name. So we can recycle things. So Glossopetalon spinescens, variety spinescens is the type for the genus. And this one, you really don't see any leaves for most of the time that you encounter it. So you might see some leaves, but mostly what's going on is there's photosynthesis happening in the stem. So they'll stay green throughout the growing season, but a lot of times they drop their leaves pretty quick. So they're early deciduous. Also in this group, which kind of makes sense if you're dropping your leaves, you don't have any stipules. And so this is going to be the taxon that doesn't have stipules. And then here, this is a picture of one of my collection sites. That's El Paso down there. And so again, we see these rocks again. They love, love, love cliffs and beautiful rocks. And so you'll see this down in southern New Mexico. And then I need to do some more sampling throughout New Mexico to see the distribution statewide of these varieties. But to make a broad generalization based on what I know, southern New Mexico is probably where you're going to see this one. Okay, one of my favorites. So variety Plantarum. This particular taxon this is my representative that was collected here in New Mexico. So in my study, I do have a collection site, and it's if you get off on the exit past the Santo Domingo exit on I-25, there's like a reservoir. You see some mining off in the distance. It's off in those limestone cliffs. And this particular variety has these massive, like just large, swollen, dark petiole bases. So you can see the leaves have fallen off, but they still stay on. These ones are a little discolored because they're old, but they still stay on and they're super, super swollen. It also keeps its leaves. It's still pretty spiny, just like spinescens, but it'll keep its leaves versus that one. That one will drop its leaves pretty early, and the leaves are not as large as in aridum. And then for your last option in terms of seeing different varieties in New Mexico, that's going to be variety Meonandrum. And this one is similar to Plantarum in that it keeps its leaves, but the difference is that it has really light petiole bases. So those big, dark, swollen ones, you won't see that in this variety. They're just going to be light, not very noticeable in this one. So lots of variation in this species. But when we were in Arizona, Tina, my advisor, she brought out this herbarium voucher, and they have these really short, compact shrubs on them. She's like, this is Glossopetalon from the Babbitt Ranches. And I was like, it is? And we went out there. And so they're just so small. They're just tiny little shrubs. And here you can see those limestone here again. But they're super tiny. And the largest one we could find was only 36 centimeters tall. And so we thought, oh, that's kind of odd because those species I mentioned earlier that were endemic, those are also really short. So you can tell that widespread species of spinescens apart from the others because they tend to be short. They're growing out of the cliffs, but they don't get very tall like spinescens. And so we were thinking, well, this might be maybe a disjunct population. So it's just separated from where those ones are typically found. Or maybe it's a weird variety of spinescens. Maybe it's something new. We weren't quite sure. And then on top of its size, it was also super hairy. And you didn't really see that in a lot of the other taxa. So the leaves were super hairy. Here again, the leaf blade would be like right here. So I'll just draw that out for you. But here's the petiole. This is a special microscope image of this. And then you can see your stipules here again. They're hairy, that base is swollen and it's hairy. So this population just didn't really fit with anything that we knew about the varieties or the species description. So we were really curious to see, well, what is this thing? And then maybe is that potentially something new? And so this is where I saw Glossopetalon, and I really remembered because I wasn't distracted by beautiful red rocks. Okay, so upon seeing this, I really needed to look at, well, what do we know in terms of, are these species just descriptions of what we've seen on the landscape? And do we have any type of genetic data that support the groupings that have been made by botanists? And so, oops. So that led me to find this paper. And so you could just concentrate on this red box up here. And so this is the family that Glossopetalon is in, the Crossosomatacea. And so it's a super small family. But the only thing we really had available is one specimen representing the entire genus. And so that's all that was done. We didn't know anything about the relationships within Glossopetalon. And so that presented a great opportunity for a master's project. And then additionally, because I was already going to do work on this genus, we wanted to see if we could replicate this relationship within our family. And so here, Glossopetalon is sister to this genus Apachuria. So they share a common ancestor here at this node. They're paired here, so they're sisters. And Apachuria, you can also find this. There's only one species in it, but you can find this group in New Mexico too. So if you go down near Magdalena in the San Mateo Mountains, Apachuria likes rhyolite. And so you can find Apachuria there. For Crososoma, so we're talking about this group here, Crososoma can be found in Arizona and California. It's actually used as an ornamental in some places. And they'll grow it because it's so beautiful. It's a beautiful native plant. They have these really big petals here. And then Velascoa, this one, I don't have a picture of it, unfortunately. I just have a picture of the seed. But this one is found in the Central Plateau of Mexico. And so these ones were hypothesized or supported to be sisters to each other. And they have, Velascoa has this crazy arrow here on the seeds. I wonder what kind of dispersal goes on there. So my research questions ultimately ended up being, what are the species relationships in Glossopetalon? We have no idea at that stage. And then are there morphological characters that define the group? So we already had information, like everything I just told you about, well, this tells you, this is how you can identify this species. So I wanted to see if there were any more based on that genetic data for the relationships. And then how about the varieties? We've got 6 varieties throughout that spinescens range. Are any of those potentially species themselves? Maybe they're also endemic. What's going on with the varieties? You have all those cool morphologies that are unique to those populations. Maybe they're their own thing. And then of course, what is that crazy short, furry population that's on the Babbitt ranches? Is that one of the short species that we already have described? Is it something new or some other variety of Glossopetalon spinescens? So that's what I was trying to figure out during my master's. And so to answer these questions, I won't go too much into the methods, but to answer these questions, what I really wanted to do was, one thing that could be going on with that short population is maybe they were being eaten by cattle. So these were present on the Babbitt ranches in northern Arizona. And we did see some evidence of herbivory where it literally looked like you had something come up and take a bite out of the shrub. And also when we would be out there, a lot of other things would be dormant. And so perhaps what was happening is that Glossopetalon was putting out new leaf material. And so might have been the best thing that was available to cattle, or mule deer is out there too. Maybe little rabbits, but they looked like very large bites. And so what I did is during my master's program, we constructed these like little chicken wire houses and just put them over the shrubs. And then we left them on until after I graduated. So for a couple years, just to see, well, if we remove the opportunity for anything to eat them, will they grow bigger? And I wish I could have continued this experiment, because 2 years is honestly really short for how slow the taxa grow. But you know, you do what you can within your opportunity to go to school, etc. So we enclosed some of those guys. tried to see if, and compared them with the ones that were left unenclosed to see if, yeah, they're getting bigger. They're just being eaten. That's why they're short. And then the other thing that really hadn't been looked at in terms of traits for these different species and varieties was seeds. And so we knew that there could be different colors. So they could be tan to like a creamish to a light brown to a dark brown. But we didn't know if any of the species, for example, only had brown seeds, or if any of that was consistent across the varieties either. So I wanted to look at the seed morphology, like the seed coat. So here's that arrow and then the seed, and then the coloration too, to see, well, maybe that'll help me tease apart who is who, and then help me write a key that will help identify this group of shrubs that can be kind of difficult, honestly. Those petiole bases are helpful, but they have a lot of variation, and then the stipules can fall off. And so you have a lot of things that can make it difficult for you to identify these in the field. And so I really wanted to help with that with my work. And so in addition to the morphology, I also looked at things that were in the Flora of North America treatment for Glossopetalon, just to reassess them and see if I saw anything different. And then I for the genetic stuff, I did a bunch of DNA extractions and got genetic data. And that's what I used to construct my phylogenies. And that's what we'll go over here next. And I can certainly share my publication with you all. And it has a nice little Excel table that'll break down the morphology, and you can just walk through, you can just compare like the different traits and then which each taxon has. And I think that'll be nice for the field, but I have a key too, so you can also use that if you wanted. Okay, so here's our results. This is based on my genetic data. So this is my phylogenetic tree. And you can just think of this as a family tree for all of the taxa in the family, the Crossosomataceae. And so let me just point out here, this is all of our samples that are Glossopetalon. And so remember our sister Apachuria. So that's going to be the same sister that we saw in that other tree. So that was supported. Apachuria that we find there in the San Mateo Mountains is sister to Glossopetalon. And then Velascoa and Crososoma were also sister. But one of the important things that we try and do when we're assigning all these fancy names and coming up with what is a good taxon is we try and make sure that everyone is more closely related to each other than to other things. So Glossopetalon, they all come out from this ancestor back here on this node. They form this nice little grouping here, or clade. And so that supports that this is a good genus, right? It's a monophyletic group. They all come out from the same place, the same node. And so that was good. Now, this branch here for our family tree is Glossopetalon pungens. This is one of our endemics that's found in California and Nevada. And so because so here's our last ancestor, and then we have this lineage. And then if you follow this branch, then we have another ancestor, and we've had more lineages come out there. So this is going to be our oldest lineage in Glossopetalon. It's sister, because of this node, to everyone in here. And Glossopetalon pungens had been argued about whether or not it's actually more closely related to Apachuria, because they look pretty similar. So Glossopetalon pungens differs from the rest of the taxa in that it has terminal flowers. So here's some there, they're kind of wilty. Terminal flowers, and everyone else in the genus, instead of terminating in a flower, they actually have axillary flowers, so they'll just come out. And Apachuria does the same thing. They have terminal flowers too. And I also think that the leaves look almost like very thick and succulent-y to me. And they also, this point is like, they're very mucrinate, but the one in pungens is very elongated. But the leaves to me look kind of similar too. So I understand why people were saying, well, maybe this is actually in a Paturia versus being in Glossopetalon. But my work supports that, again, because it shares that last common ancestor is more closely related to all of the rest of Glossopetalon than a Paturia, that it is in this genus. So we have put that debate to bed. Now, for everyone else, you can see we have two major groups. So we've got a node here and a node here, and everyone else comes out from there. Everyone else is going to be Glossopetalon spinescens. And we'll talk about this a little bit more. But ultimately, what was really cool about this is if you look at everyone that's in this group and everyone that's in this group, they correspond to biogeography, so where they are throughout the species range. And so here, if you draw a line through all of our samples, if you draw this line, that everyone that's above this line, I'm calling that lineage 1, they're in this group. And then everyone that's below this line is in this group. And so people have asked me, well, Maya, why don't you just break these into 2 species instead of having all these varieties? Make it easy. Then there would be 3 species in Glossopetalon. We wouldn't have to deal with all of this stuff. But the problem is I don't have any characters. Like, how would you tell this species apart from this one? The seeds did not pan out. And so the coloration was just all over the place. It didn't seem to have any indication of what group you're looking at or nothing. So unfortunately, I wasn't able to rename these as one species and rename this as one species because I wouldn't be able to tell anyone how to tell them apart, aside from, well, if you're behind this line that I just like picked out myself, I don't even know if it's like truly real, you know, then you're in this species versus this one. So sorry, I actually made it harder, but but that was a really interesting portion of this study. And so when I come back to this, I would love to do some more sampling, especially right across this line, to see if this pattern holds up and have just more populations. And if so, maybe this corresponds to something that happened, like maybe like the Colorado River, or like something, a mountain range or something that would isolate these 2 groups, you know, in a long, long time ago during evolutionary time. We'll skip this. This just shows that it supports the same thing. Okay, so we talked about having 4 species. We had spinescens. This is that really widespread one. We had pungens, which people thought was a part of a Paturia. And then we had clochlei and we had texense. So because these are in this group, so they're more closely related to the other varieties of spinescens than, like, let's say coming out from here. That means that they're going to have to be classified as varieties. And so that's what I did in my publication. But the great thing is we have characters that can do that. So you can tell clochlei apart from Meonandrum and microphyllum, same thing with texense. And so I went ahead and proposed that. And so now these are the new names that have been assigned as of 2020, I think is when my publication came out. Maybe it was last year. For those 2 former species. And so for clochlei, this is the one that's growing straight out of those rocks. And this one's a beast. This one's near this is near Las Vegas. This one is going to be really short and matted too. And then they have incredibly small leaves as well. But they're endemic, so they're only found in that particular mountain range. And when I collected this, and I showed you that picture of my knee there, I was looking down and I was so absolutely terrified. When my advisor went back to try and get seeds for me, I was so terrified of the actual cliff that I totally forgot to tell her that you have to like crawl on your hands and knees to get through a burn area in order to get to them. And so when I gave my master's defense, she like told the audience, yeah, you know, like we were crawling on our hands and knees to get to this population. And I was like, oh yeah, I totally forgot all of that. I should have warned you. But yeah, so field trips for Glossopetalon, as long as we have some great populations that are accessible, are doable, but they can also be pretty exciting and adventurous. Okay, now for variety Tech Sensei, these ones are very big, very leafy, very much like aridum, but the difference is these ones do not have like super robust stipules coming out of their leaf petiole bases. And they also have a super thick mid vein too. There's no hair, so in this microscope image, they almost look like they're glistening. But there is no hair on these ones either. Okay, and this went away, but here is that population on the CO Bar Ranch. And so we were really curious to see where this would come out because, again, it was so furry. They were very, very short, and it's actually really, it's right in the middle of variety aridum. So that's that really widespread leafy variety that you will mostly see. And so we were like, oh man, but because it was so different, what we would have to do for the key and being able to identify it is that we would have had to really expand the key. So this is a subset of that table I was telling you about, where you have the heights, all that good stuff to tell them apart. So let's say we wanted to call this part of aridum. This would no longer reflect that population. And so what we would have to do is say, okay, variety aridum, you can have that inhabit sandstone, limestone substrates. Now variety aridum, instead of the smallest thing that you can find is 25 centimeters, now it's 5. So it ranges from 5 to 300 centimeters. It can either be erect, so like really tall, or it can be a low mounded shrub. That wouldn't change. The stipules can either be super, super, super small, or they can be large. This is the, like, your vestiture is like your skin of your leaf. I don't know if that's a good example, like how you feel, how you look and how you feel. And so, so glabrous, so you have no hair, or you have tons of hair. So that would not be helping anyone identify it, because it's like you can just be anything. And so what we ended up doing, and also because it's a variety, you have some wiggle room in assigning. It's not like you're dealing with a species with taxonomy. So you're describing variation. We know that these aren't like an actual species. They don't just mate with each other. And so you can do this kind of thing. And so what we did is we described this as a new variety called variety Goodwinii. And then that way you can just have this, and it'll identify those northern Arizonan populations. And then here's just another picture of that, the furry leaves. Okay. So in addition to that, if I could go back and tell you, this is what I learned, this is what I would do different. These boxes around the different colored points are all of my collection sites. Now, what I should have done is, as I was going to all of these sites, was take samples of aridum throughout the range, but I didn't. I only was like, okay, I got to get here, I got to sample this, then I got to go to Texas, then I got to do this. And so I don't have samples representing like the northernmost extent of the range, or like in here. So a great future direction would be to sample more robustly throughout the Western United States, and then see if aridum has some pockets of things, and just basically see how these all come out once you have more populations. Another thing that needs to be done is that sampling in northern New Mexico. I mean, in northern Mexico. So this clade here that corresponds, we're talking about these purple ones, and we're talking about this brown one, and then these pink ones that are down in here. That's what we're talking about in these, that's these samples. They don't correspond to who's closer to who in terms of who's more closely related. So even if you are geographically closer, it doesn't mean that you're more closely related in this tree. And so here, that texense, that's really important, right? Because it's an endemic species and we want to conserve our native species. Me reducing it to the variety level might have the impact of it's not prioritized as a species, right? And so there's no conservation that can be done because it's just a weird spinescence, right? We have tons of spinescens. They're not of conservation concern. And so what really needs to be done is better sampling in northern Mexico and throughout Texas to see if there's some more population structure there, and then see if once we have that, this is a lot of land, right? That's a ton of land. Once we have that, if we could have this re-elevated to its own taxon, because then you would have the infrastructure to conserve it and advocate for your native flora based on the taxonomy. Also, it would just be interesting. And then Planetarium, they're really strong. They all group together. They were great. Same thing with our samples from Meonandrum. They look alike. Remember, we both have both of these have leaves that stay on, but they differ in that stipule base, that petiole base. But yeah, so that was the rest of them. Okay, so basically what ended up happening out of this project is that we described that CO bar population, and I have other ones. I went through like all the herbarium vouchers, and so there's a lot, there's more in northern New Mexico. I mean, northern Arizona, excuse me. So we described that as a new variety, and that's Glossopetalon spinescens variety Goodwinii. We demoted Cloclei and Texense to varieties based on their genetic relationships. And then we know that these varieties, so they form two lineages. And the pattern that we saw was it's probably corresponding to biogeography, but we couldn't find any characters to make these into their own species, unfortunately, but that's okay. So we now have two species in Glossopetalon. So that's Glossopetalon pungens and Glossopetalon spinescens. And what is that? 11 varieties? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 varieties. So that's what I did. And it was supported by the Babbitt Ranches in terms of funding. I did a lot of my genetics work at NAU Genomics Lab. And then I had a wonderful supportive committee and wonderful Leela Andrews is a great geneticist, and then Ellie is awesome, and just I had a whole great group of people to work with. And so with that, I shall end my presentation, and I'll take your questions, your comments, your concerns, anything of that nature.
And I'll just say, thank you so much, Maya. Amazing, mind-boggling. And if people want to put questions in the chat, I'll see them very easily. But if you want to raise your hand and just ask a question, you can do that too. I'll look for the hands on the squares, and I think we have a little time if we want to have that kind of a discussion. And I had one question, Maya, that I'll just kick things off with. Mm-hmm. Are you aware of any uses by people of this plant, like of the sap or medicinal uses or anything like that?
The only thing that I've seen is that it was used for tuberculosis. And I don't know if that was because it was successful or if people were just desperate. So I don't know the effectiveness, but that's the only thing that I've seen so far.
And would that have been in New Mexico too? or maybe
Mm,
in indigenous times. So they didn't have tuberculosis, I don't think, until contact.
right. Yeah. And I don't remember. It was in a really old ethnobotanical book, but I don't remember if that book was a regional book, or I don't remember the specifics about where that was being used. I see Kent's hand up. Oh, and then we'll go to
There are
Eve
some, yeah,
after.
yeah. So Kent, go ahead, and then we'll look at the chat.
You're muted.
You can click a button to ask him to unmute.
Tom, are you? I'm working on it.
Okay, I'm sorry, I had my
Good.
microphone off. I was wondering, what other family or families is this Crossamalaceae related to? If you go back in time, how did it evolve into a family, and what is it related to in its evolutionary history?
Oh, that's a great question. Okay, great. Now I can pull out more slides. All right, so we'll come back here. Let me see, where were we? Right here. Oh wait, what can you see what do you see right now?
I see the flowers and the tree
Okay,
in the
great.
middle, the phylogeny tree in the middle.
Okay, so here's the Crossosomatacea, Stachyuraceae. Oh goodness, I forgot if that's in. I can't remember where this is exactly. And then I know this family is endemic to Guatemala. And then a lot of the families actually are found in Asia. And so what's also been curious based on the familial relationship, so you kind of have, and I think they actually have this on the side. Let me get back actually over here. Yeah, so here's our hemisphere that's what these lines are supposed to indicate here. So you have this crazy, if we look at this is the order here. So just another group. We love to put things in boxes. So at that level, so this ancestor, somehow we got all of this divergence, all this speciation that happened in the northern hemisphere, and then we got some in the southern hemisphere. And then what's been proposed is that this happened during, this is what if we reconstruct it, this is what we think the continents looked like during the time that divergence happened. So when we had those two groups split off from each other. And so we don't really know if where the ancestor was. So did this kind of happen in Asia? Have it go the other way? Was it simultaneous? And so that's going to be a new great question that's at a higher level, just because that disjunction, you have to have more families included. But that's an outstanding question for this group, for the order.
Yeah, thank you. What if you go back even further? I'm trying to see the basics of the tree. I mean, for instance, where did the canopods, are they related to the order, the Crossometalales? Or
Oh, I see what you're saying. Yeah, I have another tree.
another tree?
And then while I pull this up, I'll get to Eve's question. So how does dispersal happen in this group? And so we think, we're not 100% sure, but I mentioned those arrows that were on the seeds, and then the one in Velascoa was really, really crazy and like it almost looked filamentous. I've seen ants carrying them around with their pinchers, so they like latch onto that arrow and then transport them. So I think that they're ant dispersed, but we don't actually know for sure. And then here's our phylogeny that's like very, very zoomed out. And so here's Crossosomatales here. And so it's in the malvid clade within roses.
Okay,
So
so
we're
it's
right.
related to the rose group then.
Yep.
Thank
I
you.
guess, and I think, so you got Eve's question.
Mhm.
What about the question about, I'm curious if you think the hairy low growing variety is an adaptation perhaps to drought or wind or something else. And I think Yasmin came on a little bit late. You talked about grazing, but go ahead and comment.
So sheer, especially, so we look back. Oh, sorry, I didn't mean to cut you off. Okay, is unstable. It's frozen.
Yeah, just, let's wait a minute and
Am I
see it.
back?
Yeah, there you go. You're back.
Okay, great, awesome. Okay, so in terms of wind shear, I absolutely think that that's a contributor. So here we have like a drop down there, and then you can see the horizon that's like a lower elevation. So it is quite windy up here. Grazing, the only reason I think that grazing would be a factor was because it looked like things were taking bites out of them. But if I hadn't seen that, I definitely would have thought wind shear, and then also just desiccation tolerance. So drought, UV, your typical things in deserts, because we see a lot of hairy plants in deserts. And so I was hoping that, or hypothesizing that those would be genetically correlated. So you would see in that tree, you would see a lot of difference there because those traits are representative of the underlying DNA that's making them. But we didn't really find that. It kind of popped out in aridum. So there could be something else going on there, like maybe it's plasticity. So that you just have the ability to express different depending on your environment. So I do think that your selective pressures would be wind shear, UV radiation, drought, herbivory.
Yeah, thank you. The only reason I asked the question was I was just reading something last night about C3 and C4 plants and how many of them have, the ones that are adapted more to drought have the thicker leaves and hair and things like that. And the hairiness is actually an adaptation that helps with wind shear.
Mm-hmm. Yeah, so I was thinking for the wind shear was the size. And so
Yeah, it could be too, but
it grows.
I guess they were saying that, planting plants that are hairy actually can help with protection from the wind, something
Mhm.
like that. It was interesting. So
Yeah.
that's where the question came from.
Awesome. I love that you could that this kind of built on something that you were already reading. I love that.